further ado, then let me introduce Dr. Sagar Khare, who's a professor in the Department of Chemistry and Chemical Biology and a resident member of the Institute for Quantitative Biomedicine. I mentioned earlier that he's the head of the graduate program. And Sagar, I look forward to hearing your presentation. Thank you for all of your support.
Let's see, hopefully. Can you hear me and see my screen?
Yes, yes.
Okay, perfect. So thank you, Stephen, and thank you to all of you for attending. So today I'll give you a very brief introduction to the SARS-CoV-2 spike protein that you've heard or that has been alluded to in the previous two talks. And this is obviously a very important protein for this virus. And let's see. And here's a brief outline of what I'm going to tell you. So first we'll look at the spike protein and look at its structure and what it does. You can see this in perhaps the most emblematic image of this year is this image of rendering of the SARS-CoV-2 virus from the CDC. And you can see that the spike protein is like a big red flag, being waved at us by the virus. And so you can see that as these red studs, which look like triangles. Once we look at the structure and function of the spike, we'll consider what implications it may have about the infectivity of the virus. In other words, we'll try to understand if the spike structure and function has any contribution to the fact that COVID-19 seems to be so infective, especially compared to SARS-1, which is very closely related, but still this virus seems to be very different, as we heard in Steen's talk. The progression of the disease and the appearance of symptoms and infectivity is very different. And so it's fascinating to think that a small difference in the amino acid sequence can make such a huge difference in the way the virus pathology as well as transmissibility and infectivity is manifested. And then, of course, everyone is hoping and praying for therapeutics. And so we'll look at a little overview of the kind of options that are being developed for both blocking and chopping the spike protein, as well as mimicking it so that we can train the immune system to recognize it. Okay, so let's first start with a review of the viral life cycle that we just heard also from Stephen. And I would like to draw your attention in particular to this step here. So this is the virus and the spike is now in green. It recognizes a cellular receptor, which in this case is known now to be the angiotensin converting enzyme 2 on the surface of human cells. And then through a series of events involving the function of the spike protein, there is membrane fusion and therefore viral entry into the cell. And so the virus is going to enter, and then the genome is going to be released, and which is a positive strand RNA that is going to be translated into protein. And in particular, the viral polymerase will be translated, and it will go on to both replicate as well as transcribe the structural proteins and non-structural ORFs, as we also heard from Stephen. And all of these proteins, once they are made and processed by the main protease and the papain-like protease, they will all assemble together into a virion, a mature virion as it is called. And then the reverse process will happen, which is that now this membrane will fuse back and will release the virus and so on. So this is how the virus uses the host machinery to essentially replicate itself. And a key step in this process is the first one, which is the binding of the spike to the receptor and associated changes that lead to membrane fusion and the entry of the virus. So with that in mind, let's think about how that happens. So let's start simply from a very, a cartoon representation. So I said that the receptor that the spike recognizes is the ACE2 receptor, and this will be a player that we'll see a lot more of as we go through this talk. And what happens is that there's not only just one receptor, but there are other receptors involved as well. So for example, there is a co-receptor called TMPRSS2, which is involved in cleaving the spike protein or modifying the spike protein by proteolysis, and I'll explain what that means. And it serves to activate the spike. And so this kind of activation is extremely important. That enables the spike protein to then cause membrane fusion and subsequent release of the viral RNA. Okay, so let's look at that step in a little more detail. So what happens is that there's this viral spike that you see, and there is a part of the protein that binds the receptor. Once that part binds, as I said, there is another receptor either on the surface of the protein or perhaps even in the extracellular space, which can activate this spice by a modification called as proteolytic activation, which is essentially cutting the spike protein so that now it can escape. A part of the spike protein can escape that you can see here. And then once this part has escaped, now it reveals the fusion machinery. And that fusion machinery then goes and inserts itself into the host membrane, and you can see that now the two membranes have fused. And this is common to most coronaviruses and even for other viruses like influenza, that there is protein machinery that causes viral membrane fusion to the host membrane. Okay, so let's look a little bit in more detail at this spike protein. So this is a very long protein compared to the small proteins that we're used to thinking about. So this has about about 1,200 or so amino acids, and there are two subunits. So there's the so-called S1 subunit and the S2 subunit, and the S1 subunit is involved in binding to the human receptor, and the S2 subunit is involved primarily in membrane fusion. So there are a few things that I want to highlight or draw your attention to. The first is that if we just look at the one-dimensional sequence for a moment, there's this RBD, or the so-called receptor binding domain. So this is the part of the spike protein that interacts with ACE2, and it sort of docks against this ACE2 protein, and that causes then subsequent events. So clearly, if we're thinking about therapeutics, this becomes obviously a target because this is essential for the virus to enter the cell. Another important domain that I would like to draw your attention to is the fusion peptide, or FP, that is shown here. That is part of the S2 subunit, and that is involved in creating, bringing the two membranes together to cause the virus to fuse with the host cell. So those are two of the many important parts of this protein. There are two other things that I would like to draw your attention to in the sequence, and the first one is this boundary of the S1 and the S2 subunit. And this here has been labeled as S2/furin, which means that there is another enzyme or another activating enzyme, much like TMPRSS2 that we just saw in the previous slide, another enzyme called furin that is involved in similarly proteolytically cutting this part and activating the protein. And we'll think about that in a minute. Similarly, you have a second proteolytic site called the S2' site, which is where the TMPRSS2 protease is going to cleave. So these two proteolytic sites are very important for the life cycle of the virus and for the function of this spike protein. So let's see how this protein is going to look like in 3 dimensions, right? This is just a 1-dimensional view, but in the bottom, you can see that this protein is a trimer. So it has a cyclic 3-fold symmetry. So it's made of 3 identical subunits that arrange themselves at the vertices of a triangle. So you can see that in the top view much more clearly. And you can also see that it sort of looks like a flower, right? So on the top, you have the receptor binding domain, and on the bottom, which is where the viral membrane is, you have all the other parts that get exposed afterwards and cause the membrane fusion, or the S2 domain. And this S2 furin site, which will become a sort of key point of consideration for us in a minute, that is present here. And you can see that this is exposed to the solvent and located quite outside the center part of this protein, right? So, okay, so there is, this is trimeric, the receptor binding domain is up, and these proteolytic sites, especially the S1, S2 site, is solvent exposed. One thing to notice is that the receptor binding, orientation of the receptor binding domain is extremely important. So in the previous slide, what we saw was all 3 receptor binding domains were lying flat on top of the stalk. Whereas in other protein crystal structure or cryo-EM structures that were determined by a technique called cryo-electron microscopy, you can see that one of the 3 receptor binding domains is pointing up. And in fact, David Gutzel made a beautiful representation of these 2 orientations of the RBD in his molecule of the month representations, as you can see here. So this RBD is sort of like a thumb that is this pointing up, right? And it can come down and sit on the rest of the hand, or it can point up. And I just have 2 thumbs, but imagine that, there's 3 of them, right? And so these thumbs can go up and down, and there is some kind of an equilibrium between these 2 states. And this equilibrium, turns out, is going to be very important. So all the parts that bind the ACE2 receptor are in the down state are hidden away, and in the up state are now exposed, so that the RBD can now see the ACE2 receptor. So far we have talked about the top part, but remember that this is a really long protein, and it's not only protein, but there are many other things attached to it too. So here's a sort of representation of what that protein actually looks like and what it does in real life. So the things that you see here that just appeared in green are so-called glycans, or they are essentially sugars that decorate the outer surface of this particular protein. And that's why it's called a glycoprotein. And we know exactly where these sugars are going to be. And you can see that they coat the outside very well. And many viral proteins will have that, because that affords them protection from the immune system. So you'll see that there are lots of these glycans. And remember that there is a part of this that actually sits in the membrane. So this is where the membrane is. So you can see it at the bottom. And this is, of course, not a static entity. So it's probably sort of wiggling and jiggling, as shown in this movie built by these researchers. So this was the structure of the spike protein. And now let's think about how the spike protein is similar or different compared to the SARS virus, right? And so if we think about just SARS-CoV or SARS-1, both of them belong to the same group, as our boot camp participants have looked at have done some phylogenetic tree analysis, and that both of them belong to the same group, meaning that they are essentially quite closely related. And that is reflected in the high identity between their spike proteins. The receptors that both of these proteins use for cell entry are ACE2. They also have this co-receptor TMPRSS2. And yet, as we saw, SARS-CoV-2 seems to be much more infective than SARS-CoV. And by infective, we mean not only that it sort of has spread around quite a bit more, and that there is asymptomatic spreading, etc., but also it has higher tropism, as it is called. And what tropism means is the ability of this virus to enter many different types of tissues. And it seems to even enter and reproduce in tissues which have low ACE2 expression, right? So clearly, there's something about this virus that is very different in that sense, and is allowing it to infect many more tissues and also spread in a different and perhaps faster way. And so naturally, we want to look at the differences that exist between the SARS and the SARS-CoV-2. And I think you saw this slide earlier as well in Steen's talk. One of the key differences that SARS-CoV-2 has compared to SARS-CoV, as you can see here, this is SARS-CoV and CoV-2, is the existence of this polybasic cleavage site. And this is the site that furin recognizes. And these types of sites are not found among all its closely related viruses in group 2b, but they are observed in some viruses in the in its relatives. So alpha beta 2a and beta 2c groups have this kind of site. So clearly, this has jumped into this particular species or strain of virus, and this creates a cleavage site that furin can cut. And this means that it provides another means for spike activation. And one of the ideas is that perhaps this pre-cleaved spike, and I'll tell you what pre-cleaved means, will make it ready to deploy and might contribute to increased infectivity. So what do we mean by pre-cleaved, right? So I told you the life cycle of the virus at the very beginning, and I told you that there is a step at the very end, which is the packaging of the virus. And at the time of packaging, it is believed that there are certain proteases inside the cell, including furin, which can cut at their substrates and thereby actually activate the virus. So what we have is, as the virus is being released from the cell in which it is dividing, it is cut here. And that means that the S1 and S2 domains are no longer just connected by a single chain, but they're just sticking together. And you can think of this as winding up a spring, and perhaps, having activated the virus in this way, it is now ready to sort of be deployed and enter cells much more efficiently, because one of the key requirements for entry has now already been performed even before it encounters the receptor. So that could be another clue which tells us why this virus is that much more infective. So a second clue comes from looking at the binding of the receptor binding domain of the two viruses. And people have looked at their binding affinities by a variety of biophysical methods. And the general consensus seems to be that the SARS-CoV-2 receptor binding domain binds the receptor with about an order of magnitude higher affinity compared to SARS-CoV-1, even though they do look quite similar. Their sequences are different, and so which means that these subtle differences in the way that they interact with the ACE2 receptor, which you can see in this wheat color here, and the SARS-CoV-2 binding site, those differences contribute to the tighter binding affinity of the receptor binding domain. And so the third clue that may speak to why SARS-CoV-2 is more infective is the fraction of how many times the receptor binding domain is up versus down. And in a pioneering paper that was published earlier this year, the Wiesler lab at the University of Washington found that in their cryo-EM studies that approximately half of the particles that they looked at with their technique, about half of them corresponded to this state where only one of the receptor binding domains was pointing up, and the 50% were in this particular state where they were all pointing down. So which means that the RBD is kind of hiding itself before it actually attaches to ACE2. So that seems to be another difference between SARS-CoV-2 and SARS, because in SARS, that fraction of things pointing up is much higher, meaning that the up state is more stable in the SARS virus than SARS-CoV-2. So let's now put this together and make a sort of hypothesis, which was made by Shang et al. Recently in the proceedings of the National Academy of Sciences. And the idea is that the fact that you have the frequency of RBD standing up is lower in SARS-CoV-2 means that you're hiding away the epitopes which will neutralize, or the parts of the virus that the immune system can recognize and block. You're hiding them away. So the virus is being very cunning in protecting itself. But once in a while, when they open up and ACE2 happens to be around, then we know that it binds with a higher affinity, or binds tighter to its receptor. So it's going to compensate for the hidden RBD by binding tighter. And the second, and perhaps the more important factor is this pre-activation, or winding up the spring that allows enhanced entry into some types of cells. And perhaps that may be one of the reasons. I'm sure there are several reasons, and this is the contribution of the spike to the unique behavior that we see with SARS-CoV-2. So the structure and function can allow us to hypothesize and provides this kind of deep insight, I think. Okay, so obviously the $1 million question now, or perhaps billion, is can we do something about it? We know the structures, we know the functions. So can we develop some strategies for blocking the virus, or teaching our immune system how to block the virus? And the latter, of course, is going to be the more sustainable strategy in the longer term. You always want to teach a person how to fish rather than giving them the fish. But if you're dying of hunger, as we are, a fish would be nice too. So in this case, the fish would be essentially blocking the interaction between the spike protein and the receptor. And so you can imagine doing that in two ways. One is to develop monoclonal antibodies or other binding agents that can come in and block these interactions by interacting with the spike. The other approach, which is also been attempted or is being pursued so far today, is the development of soluble versions of the cell surface receptor. So the idea is that, instead of the virus trying to find the receptor, why not give a soluble receptor that will go and find the virus and will make it happy, and so it won't really go and find the receptor on the surface. Now, of course, this can have other side effects, etc. But that is also a strategy that is being pursued. What people have also done is to look at antibodies that might exist, either that can be developed synthetically, using molecular evolution techniques, or antibodies that we can isolate from individuals that have been infected with related diseases. So for example, we could look at people who recovered from SARS 17 years ago and isolate some antibodies from them and see if they can neutralize this virus. And so initially, the first antibodies that were found did bind the spike protein, but were not able to neutralize it. And interestingly, given the subtle difference between SARS and SARS-CoV-2, it is interesting to see that they actually bound the receptor binding domain of SARS-CoV-2 in a very different place. So CR3022, this is the antibody that was isolated, binds the receptor binding domain in a place that does not compromise ACE2 binding. And it does not bind only to the proteins, but it also binds to those things that were fanning out, those green tails or the glycans, the sugars on the surface of the protein. More recently, the Wiesler lab again has found antibodies that were originally derived from SARS patients that can cross-neutralize SARS-CoV-2. So again, these antibodies are binding. This is the antibody. This is the receptor binding domain. And again, they're binding in a different location, but somehow they are able to cause other changes, and people are investigating what those changes are. and that can neutralize the virus. So that's very exciting, and hopefully we can learn from these antibodies. In fact, more recently, people from Christopher Barnes in Pam Yorkman's lab at Caltech, they published, along with some colleagues from Rockefeller University, published a comprehensive analysis of convalescent plasma or antibodies derived from individuals who are recovering from COVID-19. So now this is not SARS, but this is COVID-19. And what they have found is that most convalescent plasmas do not contain high levels of neutralizing activity, but nevertheless, rare but recurring RBD-specific antibodies with potent antiviral activity were found in all individuals tested, right? So this is extremely exciting because it suggests that if we can elicit these antibodies in general, then vaccination could be broadly effective. But also, now that we have several such antibodies, including ones that bind the RBD and others that do not bind the RBD but bind elsewhere, we can use these antibodies in synthetic formats to further develop passive immunity. So these studies have just come out last week, and I think it's extremely exciting to see that these types of the pace at which things are moving in this field is just mind-blowing. So that's very exciting. In my own lab, we have been thinking about trying to go beyond just binding for passive immunization. And the question that we asked is, what if we were to go beyond binding and instead, rather than just block it, what if we were to develop reagents that allow us to irreversibly damage the viral spike protein, the viral RBD, that is the business end. And we call this strategy PICAPS, which is passive immunization with catalytic proteins. And the particular catalytic proteins that we're interested in developing are proteases, or site-selective proteases that we are programming to recognize very particular sites. So for example, sites that we know are intimately involved in binding the ACE2 receptor, and develop nickase as well as exogenase versions of these catalytic proteins. The advantage with catalytic proteins would be that they would be required in much smaller amounts compared to binding-based reagents, where if you have 100 RBDs or bad guys that you want to neutralize, you need 100 good guys to block them, or somewhere there. But with the catalytic protein, one enzyme or one catalyst can deactivate hundreds, if not thousands, of its targets. So that should lead to extremely small dosages, and we're very excited to incorporate structure-based modeling and machine learning, combining that with experiments in cells to improve and test our designs. And so for bootcamp participants, Joey, Vidur, and Ling Jun are participating in COVID-related projects in our lab. So as I said, it's always better to teach a man how to fish. And so vaccine development is sort of also going ahead with exciting pace. And most of the vaccines that are in phase 2 or 3 have to do with the spike protein. So the Moderna vaccine involves giving mRNA corresponding to the spike protein, and the University of Oxford vaccine involves delivering DNA in these adenovirus vectors from chimpanzees. And both of them have provided exciting preliminary results. And so both of them are now enrolling 10,000 or 30,000 people in their trials, respectively. And we're keeping our fingers crossed that hopefully a vaccine will come out soon. Fauci just made the announcement that it might come out sooner than not. So this is what we talked about. We first looked at the spike protein. We talked about to hunt some clues based on the biochemistry of the spike protein and its function, what implications it might have for infectivity. And then finally, we just did a brief overview of therapeutics, which involve blocking, mimicking, and chopping the protein. And so these are the people that I have to thank, people in my lab. And then you might have seen John Williams and Cody Hoop. These are postdocs in Gene Baum's lab, and they were kind enough to share some of their excellent slides with me to help me make this talk. And of course, co-organizers of the bootcamp, particularly Stephen and Christine and Chuchi, and our IT guru, Ken, and to the bootcamp participants for being so enthusiastic and participating in the bootcamp. So thank you, and I'll be happy to take any questions.